The ontology of science is going to be primarily about biology, examples from biology. And the idea is that people have been aware of biology since there were people in the natural environment, but now we have a science of biology, which is now at a very sophisticated level, although there are still things that we do not know about biology and probably will never know. For instance, we don't know how the brain works. We don't know how consciousness exists. And from the point of view of our book, we never will. Because if we knew how the brain works, then we could create general artificial intelligence. All right, so the ontology of science, this goes back about 20 years when I first started working with bioinformatics and medical informatics people. And the idea was that the standard view among philosophers at that time was that philosophers can do the philosophy of science, but they should not interfere with the work of real scientists. The philosophers are bystanders or observers, and it's insulting to suppose that philosophers could know better than scientists how to do their work. So the goal of the philosophy of science is to understand what scientists are doing, basically. And I think this rule needs to be abandoned. And I came to this conclusion when I saw what scientists were doing, because I was working closely with scientists, and what scientists were doing at the point in time when scientists started using computers, because then there was a third force in the picture. There were scientists, there were philosophers who were bystanders, and then there were computer scientists, and the computer scientists were trying to tell scientists how to do their jobs. And they made an even worse job of trying to improve science than philosophers might have done. And so I saw the ontology movement at that time as being a recipe to solve the problems caused by software people when they tried to help scientists use software in, for instance, analyzing their data. And so I think I along with other people, I think I did quite a good job there. So the way in which software people started to try and help biologists was a form of, well, you biologists, your data is much too complicated. We can't build software that will make it integratable or analyzable. So you have to do your data in this way. And that the data then was a simplified model of the biology, and that's a mistake, as we will see. All right, so I've already mentioned before, I'm sure that the reason I was part of this biomedical community was because of the Human Genome Project. The Human Genome Project created huge amounts of data, which medical people wanted to use in order to improve their treatment of data. And so we had, again, three-cornered interaction between, on the one hand, the people who had the genome data, bioinformatics people, and then on the other hand, the people who had the medical problems, the diseases that they wanted to try and cure by using new techniques, which would be tested against the genome data. And then thirdly, you had the computer people who were trying to help the medical people perform this trick. And so this is what a genome looks like. This is just a tiny fraction of the sequence of the X chromosome in Baker's yeast. So yeast is a very interesting organism. You can have, I think, 24 generations of yeast in a single day, which means you can test ways of making yeast live longer. And if a certain process or a certain gene or a certain gene product makes yeast live longer, then it might work in humans too. And then you have life, or you have eternal life. Mm-hmm. And so yeast is a very important model organism for studies in aging. because you can do it very quickly. And this is the same, but more of it. And it goes on like that. These are really gigantic bodies of really gigantic data. And so the real work is not about genes, it's about gene products. And the most important gene products are proteins. And it's proteins really where the medicine and the genomics come to a head. And so that means you have to understand protein-protein interactions. And we can collect data about protein-protein interactions in regard to humans, other organisms with diseases and without diseases. And so we can, by looking at the differences in protein-protein interactions in people with a certain disease and people who don't have a certain disease, then we can draw conclusions about the relevance of these proteins to that disease, and then potentially find drugs which will block those protein-protein interactions. And that was the dream that all the drug companies were investing in at that time. All the hotshot medical schools were investing in that time. And the dream has realized itself in a large degree. So a lot of biomedical research, and all medical research now is biomedical research. That wasn't the case when we started. All biomedical research nowadays involves using huge amounts of genome and gene product data, and very often it involves using ontologies. Now, another goal, and you can see this already with the yeast, was that we could carry out experiments on yeast or on mice or on rats or on fish, which are called model organisms, because you can breed them as models of human being. You can breed them as models of human heart disease or of human MS or of human aging. The people who don't age very quickly, the centenarians, people who live for more than 100 years. You can find out what's the gene peculiarities or the gene product peculiarities of those people and try and find ways in which you can spread the wealth around and make more people live for more than 100 years. And I was involved in efforts to use ontology for that kind of research also. So the idea of that kind of research is that you can carry out experiments with drugs on model organisms. And because of the homologies between the model organism genome and the human genome, you can use computers to work out potential consequences of using the same drugs for humans, or the same methods, or the same collections of drugs. That particular research program has not been so successful. But the research program which says that we can use data about genes and proteins and so forth in order to find new ways of identifying possible drug design for humans, that works very well. And using model organisms to understand better processes like aging has also worked very well. But creating a switch where you can do all the drug testing on non-humans and then infer using the genome data what those drugs would do for humans, that didn't work. Or it worked to a certain degree, but not to the degree that people were hoping. Now, so you have these gigantic protein-protein interaction databases, and as soon as you start looking seriously at all the proteins, you get hairballs all over the place. And so the question, and part of the reason why you get hairballs is that different people label even different organisms, different parts of organisms, different diseases of organisms, different genes, different chromosomes. They label them in different ways. And the idea of the gene ontology was that this would create more uniform labeling. And the gene ontology is not an ontology of genes, it's an ontology of gene products such as proteins, where they are located, what their function is, and what the biological consequences of exercising that function are. And well, this is an example. So we have 11 breast and 11 colorectal cancers. And in studying those 22 cases, and I'm assuming they were all of humans, you find 13,023 genes involved. Now, the gene ontology is a hugely rich database about the functions of genes in different organisms. And so it can tell you exactly what the standard functioning, which goes hand in hand with those 13,000 genes, is. It knows not all the functions of all the genes, but nearly all the functions of nearly all the genes in nearly all organisms, or organism types. And so it gives you a kind of baseline. This is how these genes will function in a healthy organism, a normal, healthy organism. And the topic of lecture 2 in half an hour or so will be what is a normal organism. And particularly a normal human being like me. And you can use that baseline then to see what genes are elevated in their expression or diminished in their expression in your 22 case studies. And then you know where to look. You already know what proteins are going to be the outcome of the functionings of those genes. And so you know what proteins are going wrong in these 22 patients. And it did. So by tracking deviations, 189 of those genes were identified as being mutated at significant frequencies and thus as providing targets for diagnostic and therapeutic intervention. That's the silver bullet of the gene ontology, and it's still the silver bullet. It's still being used. It's probably part of software packages in every major medical school hospital. And the people who use those packages probably don't know about the gene ontology. It's become part of the tooling that they use. And all of these big so-called genome-wide association studies, which study the genomic basis for height, for instance, this is what Jorps was talking about yesterday, they also use the gene ontology standardly. All right, so the gene ontology was a great success, is still a great success. My calculations tell me that something like $3 billion have been invested in the gene ontology. But those dollars were not invested in the ontology alone. Most of the money went to tagging data with the gene ontology. All right, so these are just some examples of papers. This goes back to 2005, and now you have thousands of these papers probably every day. So the gene ontology is a structured representation, a controlled vocabulary of attributes of gene products, which can be used by researchers in many different disciplines who are focused on one and the same biological reality. So people working on aging know about the relevant gene product behaviors for aging problems, and people working on cancer know about the relevant gene product behaviors for cancer patients, and the two of them can combine their data because they're both using the gene ontology. And if they weren't using the gene ontology, they would have much greater difficulty in combining their data, and probably wouldn't even know about their data, because the gene ontology provides a kind of search engine, because you know all the data which has been tagged with the gene ontology. And there are very good software tools. One of them is called Amigo, which is a Mexican joke. I'm explaining that for Jorps. And Amigo will tell you if you have a problem, for instance, you want to study pediatric diseases in female shrimp. Then you put pediatric shrimp female, and then you press the button, and it will tell you all the papers where that's discussed, all the proteins which have been shown to be involved somehow in pediatric shrimp disease, what kind of evidence there is, whether it's evidence from actual experiments or evidence which has been calculated by computers or just speculations in papers. And so you can say, I only want the real evidence. I don't want the inferred evidence and so on. And that Amigo is kept up to date. And so every paper which talks about gene products, and many papers do, the data will be extracted and put into Amigo and into the gene product database. And it's human beings who extract the data. They read the papers. And it's that which costs all the money. These are PhD biologists whose career is scientific curation. And it means you read papers and tag entries. And sometimes it's interesting. And it's a good job if you're a biologist and you want to learn more about biology. You learn so much that you then get a better job after doing that for 3 years or so. All right, we talked about this one, talked about this one. So the gene ontology is in fact 3 ontologies. There's the molecular function ontology, the cellular component ontology, and the biological process ontology. And the reason why there are 3 ontologies is because there are 3 basic questions which you have to get the answers for when you discover a new gene. Namely, why is it there? Why do we have this gene? And not all genes have identified functions. There are junk. Well, there's junk in the human organism. Then there is the cellular component where you find it, the mitochondrion or the nucleus or the intercellular and so on. And then there is the biological process, which is downstream of the exercise or realization of this molecular function. So these are 3 ontologies, and they are 3 ontologies on 3 levels, which we will discuss later on. Yes.
The biological process, is that the cellular level? Or?
See, it's let's put it this way: it emphasizes the lower granularity level. We'll talk about that later. There are some places where the gene ontology talks about hosts of an infectious disease, for instance, or an immunological phenomenon, and the host there can be a person. But the gene ontology isn't about people; it's organism neutral. That's the whole point. It's a single vocabulary which can be applied across the entire tree of life, including bacteria, viruses, and fish, and so forth, embryos. All right, so this is the structure of the gene ontology. So we have cell components which are independent continuants, molecular functions which are dependent continuants, and then biological processes which are occurrence. So you might say, oh, well, the gene ontology stole this from BFO, but actually it's the other way around. BFO can be viewed, and it arose as a reverse engineering of the gene ontology into the level of generality that you associate with metaphysics in the philosophical sense. So this is the core of the gene ontology. It's also the core of BFO. And we worked it out in these 2 papers. All right, so, and now we can do a little bit more. So functions are dependent continuants, and the most important kind of function in the realm of proteins, gene products, is functioning. So we're not interested in side effects, we're not interested in random processes, we're not interested in what happens when a cell is accidentally dropped onto the floor. We're interested in what happens when a gene product is functioning. And so that's what most of the gene ontology is about. That's where it gets its value. That's why people use it, because they want to know what given proteins do. All right, so there are 3 ontologies. And in fact, you probably need 4. And as I just said, there are some places where you get organism level terms, and host is the most obvious one. But most of it is at the cellular or the molecular level. And that's reasonable because it's an ontology which is used to capture information about gene products, which are always at the molecular level. Now, there is a certain tragedy here. So it's clear to me, anyway, that not every process is the realization of a function. And I just mentioned some, so accidents and random processes, side effects, are not realizations of functions. And we'll come to see what the consequences of that are. So this is what the gene ontology provides: terms across species, across granularities. It doesn't say much about populations. It doesn't say much about organisms. It's primarily molecules and cells. Across technologies, so that's the really big thing. With the Human Genome Project came the invention of a whole new industry, a whole new collection of different kinds of technologies for analyzing, extracting, and observing for instance, the actions of gene products. And they generate huge amounts of data. So we get more and more data in more and more disciplines. And I said already that this helps biologists to focus on the same biological reality and to know that they are focused on the same biological reality and to find each other and to find each other's data. And so the first problem is, how do you use data successfully in order to understand reality? And from the philosophical perspective, which I'm going to be defending throughout this presentation and indeed throughout this whole course, the way to do that is to understand reality. In other words, you're not really interested in the data, you're interested in the reality that the data are about. And now the second problem is, how do you understand how the data relate to reality? To answer that question, you have to know where the data come from. And that means you have to know the machines used, the settings of the machines, what the personnel were trying to do with those machines, where the money came from, which can be relevant if you think there might be cheating, what samples are being used, where the samples came from, and so on. So there are dozens of things which you need to know about where the data came from. And that is where the ontology of science comes in. So we started with the gene ontology, but then a group of us, I mean, people before me were doing this, I just helped them. A group of us recognized this problem, that if you're going to use the data properly to find out about reality, you need to know where the data comes from, which means you need to know, need to have an ontology of science, of use of equipment, examinations, hypotheses, funding sources, papers, evidence, all of those things. And so this is a picture of what might be involved. I'm not going to go through it, but you see that you have to do all of these things before you get a simple graph and a picture of the genome involved, or a fragment of the genome. There is a lot of washing and staining and fragmenting and spinning in not a grinder. What's the thing that? Centrifuge. That's a better word. But I was thinking of something to do with cookery. What grinds? A grinder, I guess. All right. So the second problem: how to find out where the data come from. And this is a picture of the kind of picture that you would find from certain kind. These are other examples of the pictures that you get generated from these big genome analysis machines. And then these are the outcome. These are the graphs that are then generated by the computers. So this is the problem of data provenance. How, when you're doing high throughput experimentation data, do you get to know how the data were obtained? So you need to know what protocol was used, what the staining was, because typically you have a sample of tissue and you want to stain it so that only certain features of the tissue will be recognizable by the machines that you're going to put the tissue through, which machines you used, which settings were on the machines, and which statistical tools you use to analyze the data. And that can be a whole encyclopedia in itself, because people use different statistical tools. People who can use those tools know very well how to use them, and so they don't document what they did. And by the time you get to it, the statistical tool has been replaced by a new version, and you may not be able to find the old version that they used. So this is a very sad and bitter story in itself. But we need, if we're going to make any headway on these kinds of phenomena, we need an ontology or several ontologies. And so on the one hand, we need ontologies like the gene ontology, which tell us about the biological reality. And then on the other hand, we want to have ontologies which tell us about the evidence that we get to support particular theses about the biological reality. And we need to annotate our data in terms of both. And that's what people do, although it's still this which is the most common and the most impressive axis. But this axis is, from a philosophical point of view, no less interesting. So there were three proposals. The first two I was not involved in. I respect the authors of the first one. I'm very puzzled about the authors of this one. Both of those died. And this one, I was involved in this one from the start, but for the reason I named it. So the things which preceded it, I wasn't involved with because they had different names. But they died also because they were replaced by this. I was good at naming ontologies. And if I'd been there when the gene ontology was originally created, which was in a pub in Montreal by a fly biologist, a fish biologist, a mouse biologist, and a human biologist. All of you whom I knew very well, and some of them are not here anymore. All right. So if I'd been there in that pub, I would have told them it's not a gene ontology. It's something like a gene product ontology, which is less sexy. They would have made less headway with that non-sexy name, but that's really what it is. It's not a gene ontology. We still don't have a gene ontology. All right, so Soldatova and King, they are still around and they're still doing interesting work. And as I say, I respect what they do. What they do is they try and create computational laboratories or robot laboratories. They want laboratories where robots can do all the work, including working out the protocols, the hypotheses, the settings, and creating the publications for all I know. And that sort of thing never works, but it's good that there are people like Soldatova who try and do it. The reason why it doesn't work, you can read about in our book on AI. But they were working on robot experimentation, and so they needed an ontology of experimentation, and so they created something which is properly named. It's the ontology of experiments. They could have called it the experiment ontology. That would also have been a proper naming of this ontology. Because the naming of an ontology should be a guide which will allow people to find it. Because ontologies are only any good if they're used by thousands of people. They're like telephone networks. You might have a telephone network, it's fantastic. But if you don't have any subscribers, you don't have anything. And so ontologies need subscribers. All right, so Expo tried to formalize science. They were doing the philosophy of science, but with a very, very formal ontological approach. So for all the reasons which I've talked about. And this is what it looks like. And this isn't all of it, but it's pretty representative. And it may even be all of it. I can't remember. So this is the top node, which is scientific experiment. And then they have the arrows pointing in what, to my mind, is the wrong direction. So just switch the arrows. PO means part of. So this says the experimental goal is part of the scientific experiment. And I can't remember what AO is, but nearly everything here is PO anyway. So the result error is part of the experimental result. The experimental conclusion is part of the experimental result. The research hypothesis is part of the experimental hypothesis. No, that's not a part of. I don't know what that is. The representation style is part of the experimental hypothesis. Text is part of the text is somehow related to the representation style. The linguistic expression is part of the experimental hypothesis. And the linguistic expression is somehow related to both natural language and artificial language. So you get the idea. It's strange, it's not helpful, but she's picking on important things. And then admin info about experiment is related to the experiment, but it's not labeled, it's linked. And then admin info would include things like the title of the experiment, the idea of the experiment, the author of the experiment, the organ, and so on. And so, as I say, this picks a lot of the correct things that you would need to have in order to tag the data that you have. And then you can read it somewhat better here because the text is bigger. And here's more bigger text. And now let's look at some of it from a critical light. So we have experimental actions, part of experimental design. That's wrong. What she means is the design for the actions that should be performed in the experiment are part of the experimental design, but that's not what she says. And so and now another one, she says the representational style is part of the experimental hypothesis. Now, I don't really know what she means by representation style. Probably she means English and not Russian or something, but it's not part of the hypothesis. The hypothesis is neutral as to what language you express it in. And then here we have the same problem, experimental equipment, part of experimental design. That's just wrong. What she means is the type of experimental equipment that you're using is part of the specification of the type of experimental equipment you're using is part of the experimental design. And she makes the mistake in the places which I pointed out of confusing the specification of X with X. And this is, in philosophical terms, the word object confusion, or the use mention confusion better. So if I say swimming is healthy and it has 8 letters, then I'm causing a case of the use mention confusion. I'm both using the word swimming to say something about it, and then I'm mentioning it in order to be able to tell you something about the word swimming and not the action of swimming. They were never taught, because they never attended a philosophy course, they were never taught that the use mention confusion is a bad thing. And when you're doing ontology, it kills you. And very many ontologies before I came along the scene, throwing my weight and my English accent and my loud voice around, everybody was making use mention confusions. So a mouse, for instance, is I can't remember. I had all the examples in my mind 10 years ago, but they've all gone. Anyway, I could make some up. I'll give you one example of a slightly different confusion. There used to be, there still is a nursing ontology, and the nursing ontology has a definition of water. Water is a nursing phenomenon that has the molecular structure of H2O. Now that's wrong. Water is not a nursing phenomenon. And they changed it. They must have heard that I'd been bashing at them. I went around the world giving talks bashing ontology. All right, so it's clear that we are doing philosophy of science here. All of these things are taught when you do the philosophy of science. All right, that's the experimental ontology. It gets about 7 out of 10. This thing is, I nearly said crap. I mentioned it, I didn't use it. So microarray gene expression data ontology. It's always bad when they say data ontology because it means that they don't realize that data becomes valuable because it's about reality. And it's the reality that you want to have the ontology for, including the reality of experiments. So as you can imagine, they confuse all over the place things with data because they don't really know what kind of ontology they're building. And then it's full of circularity. So the definition of individual is, and this is a use mentioned, name of the individual organism from which biomaterial was derived. So these people don't know the bases. I don't think I have quite enough copies of this, but I made 7 copies. So anyone who does, there are 2 pages. I couldn't get my printer to print. Back to front. It's a simple introduction to what a definition is. If you go to the library and you look up logic of definition, or the logic of definition, you'll find big books published in the late 19th century and the early 20th century about how you build definitions. They're probably too long. That's 2 pages long. And it tells you what a definition is and how to make a definition with some examples. And one of the rules which follows from the knowing what a definition is that a definition should not be circular. Now, this is not quite circular. What they're doing is defining what this particular term, namely individual, is doing when we use the ontology. This is a reference to an individual organism. This is a reference to a name. But the problem is that they're using as the term something which is used without the capital in the definition. And so unless you understand the definition, then you will never understand the word that's defined. And then we have a similar problem here. It's just circularity. They use the word experiment twice. They use bioassay, which is a kind of experiment. And so then the experiment, they say, is all the bioassays, they're only doing microarray experiments. So all the assays and their descriptions performed as an experiment for a common purpose. And then they say an experiment will be often equivalent to a publication, which is, again, a confusion in their mind. It's not equivalent to a publication. The beginning to and the end, or the progress from protocol hypothesis to publishing the publication will be the temporal stretch of the experiment. But that's not the correct way of saying it. So that I don't really have much positive to say about MJED. It was created by computer scientists who thought that if you have a beautiful picture like this, then you have a good ontology. But if you have a beautiful picture like that, it's almost certainly a mark that you're doing something wrong in your ontology. All right. Now, there is a longer paper called Guidelines for Writing Definitions in Ontology, which you can find on the web here. So all my papers are on Phil Papers. And you should be using Phil Papers a lot because it's a good source of information and of original texts. This goes into great detail about how you write definitions for ontologies. That just tells you what a definition is. Oh, yes, here's more MJED. A chromosome is a biological sequence that can be placed on an array. Now, a biological sequence, it's ambiguous as between a molecular entity, a certain patterned set of molecules, and an information entity, namely a sequence of letters of the alphabet, which represents the sequence on the tray or on the array. Now, 90% of people are going to find something very strange about the idea that you can put a biological sequence on an array. And then they have 2 definitions, of course. That's another mark of a bad ontology. 2 definitions for chromosome. The other one is that it's an abstraction used for annotation. Now, every definition has to work in both directions. Everything which is a chromosome has to be one of these things, which could very well be true. So it's a necessary condition for being a chromosome in the information sequence sense, that it's an abstraction used for annotation, but it's not the other way around. If I want to annotate information about the jackets in this room, and I make an abstraction of jacket, by which I mean jacket in this room, then I am creating a chromosome. And something about jackets in this room is never going to be a chromosome. And so they haven't read these 2 pages because they never did a philosophy course. All right, so parallel to MJED was a rather better thing called FUGO. And did I mention this earlier? FUGO. Yeah, I probably mentioned it. So I think I said that during this era there was they wanted witty acronyms for their ontologies, and Japanese wrestling sumo is the oh, you just need a word ending with O, and Japanese has lots of those words. Yes.
Are you saying that the definitions are necessary?
Yeah, exactly. A definition is a set of individually necessary and jointly sufficient conditions for being an instance of the word you're trying to define. And that's called the INJS. So there's an acronym, but I can't remember. All right, so IFUGO was in existence. I was invited, for whatever reason, to a FUGO meeting, which was a meeting of the usual suspects, gene ontology people who were doing functional genomics investigations. And so we talked for a while about FUGO, and I said, we have to stop this because everything we're talking about applies to all investigations, all biomedical investigations. And so I told them to rename it, and they agreed, and they never looked back. And the purpose is to do what MJED and the experiment ontology we're trying to do, namely provide a control vocabulary for describing components of biomedical investigations, including design, protocol, and so forth. So the assumption I was making, which is the assumption I've been making for more than 20 years, is that you can get a long way by drawing on logical and philosophical principles from philosophy that we learn when we do metaphysics, for instance, or when we do logic. For instance, to avoid use-mention confusions, we can get very far in creating good ontologies by following some of those principles. And now these are all the communities who were collaborating on building OBI. So there were plant science communities, environmental genomics communities, standards consortium. The HUPO is the International Protein Nomenclature Standardization Body. There was IMPORT, which I worked for some years. So it's a database of patient data from clinical trials about immune diseases. And the International Society for Analytical Cytology, another standards initiative for metabolomics, the Neurogenetics Biomedical Informatics Research Network, or BIRN, which is another witty acronym. BIRN is the German for pair. And then Nutrigenomics, MJED, and the toxicogenomics is a good one. That's poison. What happens at the genomic and molecular cellular level when you are poisoned, and many drugs, of course, have a problem avoiding poisoning. I mean, in the early stages, anyway. All right, so it was very influential. It's now somewhat specialized. There are probably still as many people using it, but there are not so many standards bodies who are working on building it, partly because it's pretty much built. Like the gene ontology, it grows, but it grows rather slowly because it's reached a point where it's said all the low-hanging fruit. And now you can see that the bodies involved were heterogeneous, and that was deliberate. So the National Cancer Institute was involved. Deliberate because we wanted to cover the whole space of biomedical investigations. And it would have ended up including cancer terminology or cancer experiment terminology even if the National Cancer Institute was involved. But the National Cancer Institute wanted to have its way of defining cancer terminology represented when we were working on building OBI. And so there were different branches. There was the biomaterials branch, the data, so that's about samples. The data transformation branch, that's about taking data and processing it to create more data, but the more data is the useful, interesting stuff. And this is a general feature of all experiments. You have an input, you do something, you get an output. Sometimes the input is a material, sometimes it's a video, sometimes it's data from a database. The output is sometimes the material, but it's been scrunched or something's been done to it, spin. But sometimes the output is just data. And so all of those possible combinations are included within the general OBI view. And then we had a group devoting itself to the digital entity and non-realizable information entity phenomena. Chris Tucker is a friend of mine, and he was influenced by BFO from the very beginning. And so that has a lot to do with BFO, but I've forgotten precisely what his interest was. Bill Bug was interested in functions of equipment. For instance, we have to be able to say what a given kind of gene processing machine is there for. Why would we use it? And then instruments, plans, protocols, qualities, that's a hard one. So qualities means things like alive or dead, has 2 legs, has 3 legs, and so on. And we still don't have a good quality ontology in biology. And then roles. So again, that's the influence of OBI at work. So the roles would be the funder, the PI, the lab slave, the data collector, and then relation. So part of and so forth. And so this simultaneously with this, when this list was created, they created it on a single slide with the names of the branches, the names of the leaders. So that's Alan Rottenberg, who is still here in Buffalo. No one else here is still in Buffalo. You see how I managed to get everybody to do my work for me. This is BFO here, and this is the top level of OBI. And so that top level corresponds roughly to the list of the branches. All right, so we've got BFO, we've got function and side effect. And this is the BIRN. These are some of the targets. This is the BIRN function branch report. So BIRN was run by a man called Bill Bug, who died just before the first publication came out. He was really good. And so these are some of the functions that were worked out by OBI. So remember, the gene ontology is talking about molecular functions only. But when you're doing biomedicine, you need to know about the function of the heart, or the function of reproduction, or function of a hammer, or the function of a computer program. And so Bill Bug created a long list of relevant functions. Functions were realizable entities, which means that they can function, they can be realized in functionings. This was a very influential set of proposals from Bill Bug. So it convinced people that the OBI approach and the BFO approach were going to be useful. All right, so then if we look at functions, then we have to distinguish functions from roles, have to distinguish functions from qualities. We have to understand the relation between plans and protocols. We have to understand the relation between instruments, molecules, and cells and organisms, according to which of these serve as input. If we're interested in an experiment, then we have to understand the relation between the protocol and the protocol application, which is creating the actual process, which may be an assay process, which is the experiment itself. or we may be interested in the process of cells dying because that's the object of the process. So the assay is the purpose of the process, and the experiment will be an assay then. But the purpose of the process in a wider sense is to understand this and this and this about cell death. And then you add in more details, and that's what this is. And yet more details. So Bill Bug could go through different experiments and create charts like this for each one. All right, and then so these are the institutional entities that we have to name because they're all relevant. So the teams themselves, the funding agencies, who may have vetted the protocol for ethical reasons or other reasons, the regulatory bodies that the funding agencies have to be careful to absorb the recommendations or requirements of, the IRBs, which are checking that the team of researchers are going to do their research ethically and properly, the vendors of equipment, the manufacturers of equipment, the types of the reason you need both is because you may have to get information from the manufacturer. And then one of the projects that we planned with OVI was that we would get the manufacturers of biomedical experimental equipment to use OBI when they were publishing their catalogs. We never convinced them, partly because that idea was too sophisticated for them, partly because OBI was still quite new, partly because it would mean that you could much more easily compare the equipment created by company A with the equipment created by company B, and they maybe didn't want that comparison to be easy to make. All right, so then we need, because we have these things, we need an ontology of organizations. We need an ontology of the information entities, like footnotes, which are the outputs of an experiment. And at this point, along with Alan Rottenberg, so there are, in the history of BFO, 5 important people, one of whom is Alan Rottenberg. I think he came in to one of our meetings earlier on. He has a lot of facial hair. And then one is Werner Koisters, who is also here in Buffalo in the medical school. And if you get a chance to attend one of his classes, you will hate it, but afterwards you'll think it was fantastic because he really makes you work. And then one of them is an American who had made the mistake of marrying a French girl. She had 4 kids and then ran away. Mm-hmm. And so his career was at an end, really, because he had to look after the kids. And Pierre Grenant. And I think that's about it. They are the main. Anyway, so I said, it's not the job of biologists to create an ontology of information entities, because these types of entities many of them are going to be found in every domain, including politics and journalism and other sciences and so forth. So we created the information entity. It's called the information artifact ontology. And it contains terms like this, as well as terms like this. And so I won't mention this. Oh, yes. So we had competitors here, too. I won't mention these competitors, but there's something called the Ubiquity Publication Ontology. And so according to Ubiquity, every publication is a string. A string, in other words, a long list of characters. So a chapter is a string, and that just seems quite wrong. And so this is no good. This is more of the ubiquity ontology. It died, of course. So now if we want to know what is a protocol, it doesn't appear anywhere here. This was the first big change in BFO. So we started in 2004 or thereabouts. And I can't remember what year this is, but it's probably 2007. We made a big change because of this question. The screen is the turning point in BFO. There have been, yes.
Oh, I was just curious how one would how one would express like images if articles are just doing up characters.
Ah, yes. Well, they didn't think about that.
Oh.
Although the image would probably just be an IRI or a URI, which would point to the image, so they would get around it that way. All right, so the turning point came about when we realized that there is no place in BFO for things like protocols or for any of these things, names, addresses, and so on. So they're not objects, they're not functions, they're not qualities, they're not processes. And so where do we put information artifacts? Well, they're not strings. That was clear to us, although strings are information artifacts, but protocols are not strings. Protocols are embedded in a very complicated institutional context, including things like authors and teams of researchers and so forth. Particularly if we're talking about published protocols. And another related issue is the issue of the distinction between types and instances. So John Kearns is an instance of the type human being. War and Peace is an instance of the type novel. This copy of War and Peace is an instance of the type book. War and Peace is not an instance of the type book. Now, in understanding this here, we also understood what we need in order to be able to deal with information entities in BFO. And so with another related problem, UniProt is a big worldwide successful database of protein data. And so we have UniProt. It's a thing. People work for it. They get paid by it. Is it a type or an instance? So there are. I'll take that back. No one gets paid by UniProt because it's a database. Some people get paid by the organization which maintains UniProt. UniProt itself is a big, successful database. In principle, I could have a copy of UniProt on my laptop. And so there are lots of instances of UniProt, therefore UniProt must be a type. But we didn't like that. Me and Alan, this is all me and Alan, or Alan and I. We didn't like it because we didn't want to have types which were massively arbitrary. So War and Peace is not a type. War and Peace is an instance. It's an instance of the type novel. Novel is a type. And the reason for this prejudice is because in the history of metaphysics, types have typically been simple. You can formulate simple definitions of them. So red is a type, flat is a type, table is a type, gene is a type, cell is a type. But the pattern created by the entire string of letters in War and Peace is not a type. It must be an instance. It's an instance of the type sequence. And so UniProt similarly cannot be a type. There are many copies of the database, which is UniProt, but UniProt itself is not a database instance, and it's not a database type. I don't know what the type is. Maybe we wrote it down here. We need a word like novel, which would be, yes.
Can only types have definitions?
No, we can. That's a really long subject. So when we have a proper break, then you can come back with that, and I'll say something. But it's a good question, but it's not. It's not going to help me here. So UniProt is not an instance. Sorry. UniProt is an instance. It's an instance of the type database. That's what I wanted to say. And we have many copies of UniProt, but none of those is identical to UniProt. UniProt is what they all have in common, and you get copies of UniProt on your laptop, just in the same way as you can buy a copy of War and Peace. That doesn't mean you buy the novel; you buy a copy of the novel. All right, so that led us to introduce the idea of generically dependent continuance. And the idea is that an information entity is a generically dependent continuant. That means it's a dependent entity, but it's not a specifically dependent entity. A specifically dependent entity requires a specific bearer. And if that specific bearer goes out of existence, then the dependent entity goes out of existence. A generically dependent entity requires a bearer, but it doesn't have to be the same bearer. So you can copy Uniprot onto another laptop. You can print War and Peace in another language on other pieces of paper. And so that's a generically dependent continuant. A generically dependent continuant is a dependent continuant which is dependent upon some entity or other, and perhaps on many entities. Yes?
Also, then are you categorizing films as, or movies as generically dependent?
No, movies, that's a long story. So we talked about musical works over the weekend a little bit. So if you buy a movie, in the good old days, it was easier because you would buy a CD, and that's a continuum. The next step is to say that the movie in the cinema is actually it's software too. It's probably downloaded from the web, but it's a continuum. It's a piece of software or a long piece of data, which when you put it in the machine in the cinema, it will display the movie. So we have two things: the movie, which is, let's say, a pattern of software, and then we have a performance of the movie, which is what happens when you run the software. It creates these patterns of light on screen. So that is a process, but the movie itself is not a process. It's a generically dependent continuum.
Can you say something analogous about reading as well? Because
Yes, so this Ingarden, I didn't mention this aspect of Ingarden yesterday, but Ingarden's theory of the literary work is built around readings. So the work exists and it gets read, and gradually it gets read by different people. And as the different people introduce new interpretations or a new way of pronouncing the words, then the work itself will change, because the literary works have lives. Literary works are generically dependent continuance. Readings are processes. And oh, it's my parking thing that's telling me I have another hour and a half. All right. So now generically dependent continuant was a new term in BFO. Information content entity is the main term of the information artifact ontology. It's not a term in BFO, but it's made possible to build the information artifact ontology because we added this term into BFO. And then we have realizables. Functions are realizables. Roles are realizables. And these are some examples. There are non-realizable information entities. There are realizable information entities such as shared plans, for instance. And there are digital entities which are realizable, for instance, software which can be executed on a computer. And I won't go through, so there are more information entities relevant to OB. And in all of this, we were following the same methodology of ontological realism. We've worked out what the information artifact world is like as best we could, and we're still working on it. The IAO ontology is still very much a work in progress, partly because it was built by biologists for biologists, and so the terms that are used in IAO are too heavily influenced by biological expectations of how the definitions should look. And so my prediction is that there will in due course be multiple IAOs, which will involve the same basic terms, but which will be developed to cope with the terminology of specific communities. Yes?
if ontological realism is the goal for the pollution that we take, is it possible to, so is it like okay for oncologists to have like conventional categorization? So maybe, what some might think that conventional categorization is like not real in the world and stuff.
Give me an example.
So I was thinking of how species, it would be really hard to make an ontologic species, like biological species.
Ah.
Because there are certain,
Yeah.
especially for neurotypes, I forget. But
Yeah.
it's very conventional. Like,
Yeah, so there is something called the NCBI taxonomy, the National Center for Biomedical, I think, for Biological Information taxonomy, which does what you're doing, but it's not an ontology, it's a taxonomy. It's very big, it changes a lot because new species are identified and because some kinds of species evolve very quickly, like viruses, for instance. But we use the NCBI taxonomy as part of our suite of ontology because we need species terms. But you're right, it's hard to build an ontology. It's hard to find definitions for many of these species. Many of the species at the lower level are defined by the corresponding gene sequence or by a corresponding sequence. Yep.
Would like investing all the time into like an ontology of like species like have diminished returns if you could just have a taxonomy as the resource for like what you're doing now?
So the let's see what's the whether you want an ontology for a given domain is always a question of what you need it for and how much money you have to build it, or how many willing helpers you have to build it. And so there's no general rule here. If it became really important to have a taxonomy of eukaryotes, I guess that would be pretty easy because they're well behaved and they don't evolve very quickly. But a taxonomy is good enough. So, but if you needed an ontology, which means you need definitions and axioms and so forth, then you would have to get the funding, but you could build one. So it's that question arises with every ontology because you have to know when to stop, because there are always new terms that you can add or new axioms that you can experiment with, and you have to know, and the way you know is when the money runs, or when people lose interest, or when you already have an ontology that can perform the use that you wanted it for.
The taxonomy.
Taxonomy. So sometimes, but I have been involved recently in planning how to update a really big government taxonomy, and they came to me because they figure that the next update of this is going to have to be an ontology because they need more logical power than they get from just a taxonomy. A taxonomy is just a tree. It's a gigantic tree. Each node is a label or term, and every label is a subcase of. Sorry, every label refers to a subcase of the thing which is labeled above. That's it. It's a tree. And every ontology has to have a taxonomy as its backbone tree or hierarchy. But ontologies have more. They have more logic, they have more definition, and they have relations other than ISA. So all the relations in the taxonomy are ISA relations. So a cat is a mammal. Reproduce that 1,000 or 1,000,000 times and you get the NCBI taxonomy. They also give you numeric codes for the species. All right. So that's the end of the first part. Second part is called the canonical life, and it draws on a lot of the things that we've just been talking about. Rob Arp was one of the authors of the BFO book published by MIT Press nearly 10 years ago now, 8 years ago. And he and I published a short piece on functions. It doesn't correspond to the definition of function that we develop now. It's taken from a piece by Ross and Mechino, who were the authors of the foundational model of anatomy, which is this really great ontology of anatomy, which I talked about yesterday. They define a biological function. They define it using BFO terms. So Cornelius Ross had already adopted the BFO gospel. So it's a function is a biological function is a function which inheres in an organism, basically, or in a part of an organism, and exists and has the physical structure it has as a result of the coordinated expression of that organism's structural genes. So this is a much more biology-specific definition of function than the one which we have in BFO, but it points to a very important feature of all of the parts of your bodies. Each part of your body came into existence because you have structural genes which are already present and folded inside your the chromosomes that were present in when you were just a fetus. And those structural genes then create your spleen and your eyes and your nasal cavity. Everything is a product of these structural genes. And we have those structural genes because we need to be able to swallow and see and so forth. And so the structural genes are responsible for the existence of the functional parts, with the exercise of whose functions is seeing or swallowing or any of those other things. But now we live. Living is not the expression of a function. Living is the expression of all of those functions in a very beautiful, orchestrated, coordinated way. But we do have a life. And I want to talk now about the life of the organism. And so this comes in because we sometimes consider the attempt to develop all of these ontologies as a way of solving the Tower of Babel problem, which arose because the Human Genome Project was so successful and people were inventing new codes all the time. And this is the National Center for Biomedical Ontology, which paid for me to go around the world for, I think, 15 years, going to meetings, boot camps, tutorials, workshops of all kinds, 109 such events, where my job was to teach people how to build ontology. And so these are some of the disciplines which the ontologies then, which arose out of the Human Genome Project, for which we had to provide ontological support. These are the 3 questions which I talked about earlier that you need to answer whenever you identify a new gene product. And then the question is, how do we create the broad coverage labeling system with logical definitions, which means that they have a semantic content across the whole of biomedicine. And to do this, we created the OBO Foundry. So the gene ontology was there. Lots of people were building ontologies to carry off the same trick. The anatomy ontology is an example. But they were building them ad hoc, and they weren't following the same principles as the gene ontology. And they were very often making a mess of things. And this is where I came into the picture. So I was responsible for creating a set of principles for building ontologies around the gene ontology. And so there was a meeting where some of the same people who had been present in the bar in Montreal, along with the people who worked for them, and I presented this next few slides as my introduction to what they needed to do. And it was a great success. So none of the ontologies in the OBO Foundry is as successful as the gene ontology, but the foundry itself is still going strong. There are lots of people who work to keep it going, and there are lots of users of this ontology, and it's become a kind of standard. So now the U.S. Army builds its ontologies following these kinds of principles, for instance. So this is the OBO foundry, one of the original presentations of it. So the cell ontology, the Kebi ontology, which is a chemistry ontology, the two anatomy ontologies, one for humans and one for all organisms. Fugo was present in this first iteration, the gene ontology, the quality ontology, which is very problematic still, the protein ontology, the relation ontology, the RNA ontology, which never got off the ground. So we have a stub at the beginning, and then the sequence ontology. These were all ontologies which were present at this meeting and who contributed to the design and to the principles of OBO. OBO means open biomedical ontologies, and now it means open biological and biomedical ontology. And so we've seen this. So we have three gene ontologies. We have molecular function ontology, cellular component ontology, biological process. And processes cover all processes in biology in principle, not just those at the molecular or cellular level, but also at the organism level. We've seen this already earlier, but we've also seen that they're primarily processes at the fine grain. So now we have a granularity gulf. So most of these hotshot data sources which transformed biomedicine are data sources about molecules and cells, where the clinical phenomena are either about human beings or about limbs or about organs, or they are about all of those things, because many diseases cover, well, all diseases actually cover multiple granularities. So you have a headache, which covers your head, and then it covers all the nerves linking your head to your, your head is already linked to your head. But it covers all the nerves which form the relevant parts of what is called the nociceptive system, which is the system through which your brain identifies tissue damage. And when it identifies tissue damage, it creates signals called pain. Now, there's a lot going on there. There are the nerves, the synapses, a very complicated system of nerves and synapses, together with the conscious experiences of pain, which we still don't understand, and together with the tissue damage, whatever it is which is causing your pain. And so disease studies, which is to say medicine, is going to have ontological sources which deal with the big stuff, as well as with the small stuff, the cells, the synapses, the proteins, and so forth. And the gene ontology gives us a start. So the gene ontology, as I say, focuses on cellular and tissue, small tissue level processes, but we need to have proper attention being paid to organism level, which includes tissue level and organ level and arms and legs level and so on. But then we need the organism itself. So we need not only molecules and cells and cellular components, we also need organisms. This is how I built the OBO Foundry, by doing this. And they were pleased. They'd never seen anything like this before. And now we come to functions. And so we have molecular functions here. I wanted to have cellular functions in the organism level. I didn't want to have organism functions because I don't think organisms have functions. But many biologists say that their function is to survive and reproduce. But we can leave that issue aside. I wanted to have functions at all levels. And so then we would have 9 ontologies. We would have 3 continuant, independent continuant level ontologies, 3 occurrent level ontologies, and 3 function level ontologies. So 9. And now there is in biology a resistance to what is called teleology. So modern biology doesn't believe that anything is there for any reason. So there are no final causes. So the way to understand this is to say, well, you have a virus, the virus is doing you harm, but the virus is prospering. And so you can't really say that the virus is a bad thing or a good thing, because it's a bad thing from your perspective, but who are you? You're just an organism. So biologists are, modern biologists, modern scientific biologists are, from their beginnings of their training onwards, immunized against teleological thinking. And I'm not. I like functions, and I think that most of the interesting things in the world come about because lots of things like violins, for instance, and concert halls and tables in restaurants have functions. And so I try to usher a new age of teleology into biology. And starting with molecular functions, then I have cellular functions, then I have organism functions. Why not also functions of tables or functions of, which are easier, actually, functions of computers? Well, we didn't need to go down that road because this was biology. But around about the time where I started my argument that we should take functions seriously, the gene ontology in negotiations with the NLM, that's the National Library of Medicine, which is responsible for this kind of activity within the National Institutes of Health. The gene ontology, I won't say they made a deal. I think that they had a negotiation, and the upshot of the negotiation was that a new definition of function was introduced into the gene ontology, according to which a function is just an activity. Now, the rationale that the NLM gave for seeing functions as just activities was that people couldn't understand this fancy function talk, and activity is a much easier word to understand. And so this is what I wanted. And so I wanted some processes to be realizations of functions. And for some processes to be realizations of functions, we need to have some processes which are not realizations of functions. And then we have teleology. We have some parts of the biological, whatever it is, which are exercises of function, and then we have some parts which are not. So these are the parts which are not, and I've mentioned some examples already today. And so if you have functionings, then you have malfunctioning, and most disease is caused by malfunctioning. So the existence of malfunctioning is indispensable if you're going to understand disease, and if you're going to understand what doctors do. They try to rectify parts of the body which are malfunctioning. And if you're going to have malfunctioning, you have to have functioning. All right, so this is what I wanted. I wanted a molecular process ontology, and then a sub part of this, which would be the molecular functioning ontology, alongside the molecular function ontology. But the anti-teleologists made a very good point, which would be that the functioning ontology is just going to be it's going to repeat the molecular function ontology. Just as you have a pump whose function is to pump, so you're going to have a leg whose function is to walk. Or no, that's not right. Let me get this right. So you're going to have a liver whose function is to perform liver activity. They feared. So they didn't want to have a replication. I agreed. I didn't describe the nature of the replication very intelligently, but trust me, you end up with a lot of repeats if you try and do this. And so we ended up with something like this. And molecular functions are still there. They're still called molecular functions. The ontology is still called a molecular function ontology. But 99% of the terms in the molecular function ontology have been relabeled to be activities. And so I lost that particular battle. I won a lot of other battles, but I lost that one. And I'm still arguing in favor of my original position. So I published a chapter in a book on medical ontologies and taxonomies and terminologies, which is all about why gene ontology should do justice to the fact that cells and organisms have functions. All right. So we don't need to go through this. So that now we come back to the canonical life. So there are parts of our organism which bring about our death. Is this their function? If you're a teleologist, then you think that the teleology is going to bring about only good things. And so oncogenes bring about bad things. They bring about cancer. And so oncogenes do not have a function. And that is actually the correct biological account. Oncogenes are always genes which have a function, but they become mutated. And that's why we call them oncogenes. They're not oncogenes ab initio. So the general line is that those parts of the organism which bring about aging processes are not functional. They are, I wouldn't say functioning, they're acting in the way that they're acting because they've been mutated one way or another, or something's been mutated. But there are parts of tumors which are involved in bringing about the expansion of the tumor. And it may be reasonable, I'm not committed to this one way or the other, but it may be reasonable to say that those parts of the tumor are functioning and they have functions, namely to keep the tumor alive. And the tumors, when they reach a certain point, have entire blood systems. They have circulation of blood. And those things function in just the same way that the blood circulatory system of a healthy organism function. So, but that's a side question. A more interesting question is reproductive organisms, so organs. So there are many organism types where the exercise of reproduction brings about the death of the male, typically. So are we going to say that reproductive organs which bring about the death of the male don't have a function? Surely not, because reproduction is one of the main features of all life. Yes?
So about a function of tumor. So doesn't it has to do with doesn't it have to do with like individuation? Because I heard that the bigger mammals like whales don't have cancer because they grow super cancers that kills cancer cells.
Yep.
So there is like this competition between two functions.
Yes.
That
Good.
is like ultimately benefits the organism.
Yeah, so what you're saying now tends to support the line that biology should not allow taxonomy, because you can always have some bigger growth which kills a tumor, and maybe we will be able to bring that about. So that I don't know that there is active research at the moment to create super tumors which go and kill tumors.
I was just thinking about individuation of different as functions, because like some functions can be multiply as a whole, and then so to individuate something with their function seems kind of problematic.
Yes, so we have tended to concentrate thus far in, when I say we, I mean me. We've been tending to concentrate thus far on simple entity types. And sometimes they're very complicated internally, but the organism is a simple type. And but now we're gradually moving to study the ontology of systems. And certainly the human organism with a tumor forms a system. And the whale case that you described forms an even more interesting system. So we will be moving to a position, I think, where we can deal more reasonably with those kinds of multinodal or multi-centered biological and indeed non-biological phenomena. All right, so reproductive organs are a problem. That many of the things that you want to say about organs fail when you apply them to reproductive organs. So where biological function is determined by what contributes to the organism's survival, reproduction is going to be assessed in terms of the group's survival, or the species, or the gene indeed. So we don't need this, I think. So when we're talking about functions in biology, we're very often talking about contributing to the functioning of the whole until we reach the organism level, and then we're not dealing with functioning anymore, because organisms don't function. I am not here to do ontology. That's not my function, because organisms don't have functions. All right, so these are some examples of functions which we don't provide. And so now, function is what gives rise to normal activity. Sometimes normal activity is statistically abnormal. But still, generally speaking, whenever I've been talking about the functioning of the liver or the kidney or of any other organ or of any other organ part, I was talking about normal functioning. And but there you can have falls from the normal. So this is a normally functioning heart, and that means it's in the middle of this system of prototypes here. And then as you move out from the middle, you get poor functioning. It's still functioning, but it's functioning in a poor way. And then it's just malfunctioning. It's doing bad things, which eventually mean that you're dead. Now, so there is a great ability of functionings of functions, which correspond to deteriorations or decay or damage to the functioning part. And the doctor of the job is to fix broken body parts, to eliminate such malfunctioning or to prevent it from the start. You make sure that the patient exercises so the heart doesn't degrade. And or you could try and intervene at the molecular level by developing drugs which will keep your heart healthy. Now, the gene ontology is a canonical ontology. It does not represent illness. It only represents what they call wild type behavior, which means behavior in the jungle or in the wild, which is not behavior of a dying frog. It's behavior of an ordinary frog who's reproducing and doing the other things that frogs do. And that's true of most of the ontologies that we've developed. If we wanted to develop ontologies to cover all kinds of organisms, including organisms which have lost their arm and organisms which have lost their teeth, and so on, it would just create a monster which would be useless. And so instead we have the disease ontology, which is, and we have something called the human phenotype ontology, which is a gene-based ontology of human phenotypes, including diseases. But the gene ontology, the anatomy ontology, the DNA ontologies, and so forth, they are representing normal functioning. And so this is the official summary here. And so in this view of the ontology world, everything represented is typical. It's normal, it's not diseased, it's not a giant or a dwarf that we're talking about. It's a normal sized person with exactly 32 teeth. And then there are no giant dwarfs and there are no dwarf giants. So they're both equal in size to a normal person, but they have both gigantism and dwarfism at the same time. And I'm told that there are such people. I don't know whether the people who told me that are telling the truth. All right, so now the FMA is a canonical, which means normal representation of the types and of the relations between the types, deduced from qualitative observations of the normal human body. And then it's been refined as they do more and more observations, so that it's a really good, really big anatomy, and it corresponds to textbooks of structural anatomy. There are textbooks of surgical anatomy, and there are other kinds of textbooks. Surgical anatomy is interested in places where you can cut. But structural anatomy is interested in bones and teeth and lungs and so forth. It's an amazing, amazing achievement. But now FMA does recognize what are called variant anatomical structures. So some people have different numbers of lobes in their ears, I'm told. So they are variants. They're not abnormal. They just differ from the normal by some not common, but repeatedly occurring anatomical trait. And canonical anatomy is the anatomy of the canonical human being in the canonical anatomical position. So no lost arms, no expanded muscles because you've been using steroids, just what they call the canonical bow plan, which means the building plan. But now this is that's what the FMA is about. It's the canonical building plan for the human being. Now, this led me to speculate, well, if we can have a canonical building plan for a human being, what would be a canonical life plan? So if we wanted to do for the occurrence in the life of a human, the processes, including life, which is the process which encompasses all other processes, if we wanted to do for that what Cornelius did for the building plan, what would we get? Well, this is sort of relevant. So there are around the country specialist organizations whose job is to create model organisms, particularly mouse models. And when I say models, I mean that they are bred to create models of multiple sclerosis or models of heart disease or models of diabetes, so that you can do experiments on them with diabetes drugs to see the consequence. So we can create organisms which have a model prototypical bow plan very, very effectively, very, very impressively. So what about a canonical life plan? What would be, yes?
So when you say life, are you talking just about the biological life or like a person?
Everything.
But then if it's something that's more than just biological, like living or like,
yeah,
wouldn't it have to involve other like non-biological components like society or social?
Yes, that's the way we live, and that's the way we always live. Humans always lived in society. We don't know really, but they had to have some kind of artificial shelter mechanisms and artificial tools from a very early stage. So
I'm kind of not sure about having a canonical life.
yeah, we'll see. So this is it. You start by being born, then you're an infant, then you're a teenager, then you're an early adult, then you get older, then you're a late adult, then you die. Everyone does this. And we've not got very far. We've not got shelter and food anymore. Yes?
What's the curve?
Oh, that's a good question. I have no idea. I got this from the internet. So whatever it is, it must be true.
The population?
No, because the population depends very much on which country you look at. And Gaza, the curve would be like this, because they have more children than they have others. And it's not age either. Maybe it's impact or so. Rain. I don't think it's rain either. Some people say that males reach their peak at the age of 60. But it's not that either.
People, like what?
I think the more people you can push around, so the peak of power. All right. So now we have this idea of biological function. It it's part of an organism and has a disposition to act reliably in such a way as to contribute to the organism's survival. And then the function is this disposition. So the liver has the function of doing what it does because that contributes to the organism's survival. But we can improve it by referring to the canonical life. So an entity has a biological function means the entity is part of an organism and has a disposition to act reliably in such a way as to contribute to the organism's realization of the canonical life plan for an organism of that type. So for humans, it looks like this. And if you don't get all of this, then something is not functioning as it should. People have premature illnesses. People, so let me. All right. So this is what I'm going to experiment with, and so the canonical life plan is what is pre-programmed in the genes for an organism of this species. Just as the body parts are pre-programmed by the organism's structural genes, so there are genes which pre-program that we, for instance, reach an age where we are interested in sexual behavior, and we reach an age where we're getting tired more, and so on. Yes?
There is a function for organisms, and then life is kind of like a manifestation of that function. Do you believe that life has something use to?
No, I didn't say there is a function for organisms. I said there is a function for parts of organisms. So there, and in the future, we may be able to architect parts of organisms whose function is to make us live longer. And they do that basically by defeating cancer, because cancer is by far the most important defeatable aging process. People say.
My follow-up question is that so kind of the lost lifespan is understood what is implemented in the genes for an organism. So, but the genes, they're like goal, like like in the selfish gene.
Yeah.
Their goal is not to sustain.
Oh, I know, but I don't think the selfish gene leads to interesting speculations like this. And of course, the selfish gene would still work if we were indeed able to create genetically new body parts which made us live longer. That means that the genes would have even more impact and even more of an ability to spread themselves around. Particularly if we can continue to engage in sexual activity for longer. So I don't think there's anything that's inconsistent with the selfish gene idea here, but I just don't find that idea fruitful because it tends to leave out the organism. And I think organisms are selfish, and it's that we can understand the organism's selfishness more easily than we can understand the gene's selfishness. In fact, the very idea of gene selfishness is, in fact, just a metaphor. It's not something that a gene can't be selfish in the way that we. Yeah.
Use social insects that have organisms in specific roles.
Yes.
Their canonical life separate from the parts, like for worker ants.
Oh yes, yes, good.
Queen.
Yes.
And
So then, yeah, the canonical life for a worker ant is going to be different from the canonical life for a queen.
so you mean like
I'm going to deal with some of those kinds of cases.
Okay.
So just getting off from that question, so again, the individualization will be like a issue because it seems like it depends on like what we regard as organism to say like if they're doing economical life family before even as worker bees. I think the like the beekeepers or whatever who are into that industry, they don't regard bees or a single bee, individual bee as a organism.
Yeah, so I think that there is a lot to say on behalf of what you're pointing to now, but if we go back to this is only going to be realized if you are in a society with at least one other person of the opposite sex. Because if you're going to have, so this is going to involve having children. The canonical life plan is going to involve having children. That's not in the picture, actually, I now see. It should say early mother, being a mother or being a fatherhood. And so we are like bees, and we are tied to being in a society, unless we are lonely ontologists. But that's not a canonical life. Yes.
Oh, I was gonna say that maybe they have more than one, because that can track the lifetime of the individual, like me or you.
Yes.
You could have like me as a woman. Like,
Oh yes, of course,
I'm
of
up
course.
on stage,
Yeah,
so,
yeah.
you know, it could be like a plural, like it could be.
This
like
is-
plans,
yeah,
because you can
this
talk
is-
about me as a human, or you can talk about me as Olivia.
Yeah, this is the simplest version of the canonical lifetime, but there are many more complicated versions, which I'm going to come to. So this is true for all animals, including humans. So we have to develop from the embryonic stage. We have to grow, we have to reproduce, we have to age, and we have to die. If you don't do all of things, we're not leading a canonical life. And that applies to us, and it applies to tiny birds or worms. Another thing is that we have to have a water balance. So there are many universals which persist through our life, and one of them is water balance, and it's always at the same level. So if you drink 6 pints of beer in an evening, you will drink less the next day, and you will urinate more, because your body will always bring you back to the standard water balance level. That's what it looks like in the long run, throughout your life. And now, for non-human organisms, the only aspects which determine the